Okay, so sourmash. Today's presentation is about quick insights from sequencing data with sourmash. And we will start by skipping the first one because we are not doing Jetstream. At some point, we're going to talk about cloud resources that you can access, and Jetstream is pretty cool, but that's not today. So when you're doing sequencing data analysis, the sort of data that you get out of a sequencing center is a big file full of reads. And reads are usually sequences of characters, the nucleotides, A, C, G, and Ts. They, depending on the technology that they are using, that you're used to sequence, you'll get different sizes for these sequences. For Illumina, it's about 150 base pairs to 300 base pairs. For other things like PacBio and Nanopore, the sequence will be thousands of bases long. And there are trade-offs on how much it costs to sequence this and how many errors you're going to get in each of these sequences. But basically what one approach for analyzing this data after you have it available from the sequencing center is to, instead of treat the sequences as this variable size sequence of characters, is to break the sequences into k-mers. So the idea of a k-mer, and k here stands for a number. So a 4-mer would be 4 letters long, a 6-mer would be 6 letters long. The idea of using a k-mer is that because if you have a sequence that's longer, you can break it down into the composing k-mers for that sequence. And the way it works is that you grab the first 6, and then you do a sliding window on top of the original sequence. And then you grab the overlap in the next sequence and so on. So at first, this might sound a bit wasteful because you had something that was this size before, but now you have all these sequences that are shorter. So why would you want to do that? A big benefit is that, or we're going through there, but a big benefit is that if you're using data, for example, Illumina sequencing, where you do have errors sometimes, but most of the bases are good, they are actually real values. This will let you get runs of characters that they are going to be exactly what were in the original sequence. Like you can avoid having to deal with errors in the middle because most of the k-mers here will be valid. But the other big benefit of using k-mers for analyzing this stuff is because they are fixed size, they are 4-mers or 6-mers, you can turn that into a representation that for the computer is super fast to actually process. It's a process called hashing. And it's way easier to deal with these hash k-mers, which are regular integers for the computer, than deal with long sequences of characters that might use way more space and would be harder to index into and so on. So hashing k-mer sequences is a pretty common technique for doing genomic data analysis. And also there are some other benefits on using k-mers. There is this paper from Kozlicki and Farouche, I think, yeah, Farouche. It's called MetaPalette. That what they did is that they took sequences, they broke down into k-mers, and they analyzed based on a phylogeny to try to characterize like how long a k-mer needs to be specific for a species, a genus, or a phylum level. And what they found out, and it's pretty cool, as you increase the k-mer size, you get more specific further down the phylogenetic tree. So for example, if you have a k of 21, it's about the genus level that you can classify sequences. If you have a k of 31, you're at the species level. And if you go to k equals 51, that's strain level, because you can do strain level classification. well, they actually use for a reason for using odd number k-mers is that sequences, they have reverse complements. And if you have an even number for k, you might get the same sequence on the way back and some like the same it's a palindrome sequence. So that might confuse some analysis sometimes. So that's why you usually stick with odd numbers. So one idea for that we can use to use the k-mers to compare samples is to use one measured distance that's called the Jacquard distance. So the Jacquard distance works for any set of things that you have. And the idea being, if you have two different samples and you have some sort of set representation of those samples, the Jacquard similarity is going to be calculated by taking the k-mers that only appears in that sample and divide by the number of the union of both sets. So what does that mean is that if you when you do this division, this will go from 0 to 1. If you have 0, you don't have any similarity between both things because the union of both of them, sorry, not the union, the intersection. The intersection of them is going to be empty. So that's going to be 0. And if they are exactly the same, that's going to be a 1 because every single thing from both sets is going to be present on the other one as well. If you're dealing with k-mers, there is an issue here with genomic data analysis because the number of k-mers that you have, since we have 4 bases, A, C, G, and T, is going to be 4 to the k. That's exponential. And so if you're dealing with like 21 mers, that's 4 to the 21. And that's a lot of possible k-mers. If you're going to 51, that's even more astronomical. And that would take a lot of memory to represent all those k-mers in memory. So the trick that we are playing here is using an approach called mean hash. And what mean hash does is that it will take all the k-mers from a sample, or especially what mesh does. Mean hash is something else that mesh adapted for genomic sequences. But what the mean hash approach does is that it selects a subset of the original k-mers in a way that you can compare samples between them. And the benefit is that this subset is very small. And so it's very fast to compare them and to measure how similar they are. So it's an approach for taking a potentially very big set and find a way of getting a subset that you can compare around. So here in the lab, we have a software called sourmash. That's what we are going to go through, where we also have some other extensions to this basic idea of using mean hashes to calculate similarity between data sets. First things first, we are going to use Conda to install sourmash. So I'm going to create a new environment because that's a new analysis and I don't want to mess my other environments. And we already went through this in previous presentations. So if you want to go back and check them, they are there. So I created an empty environment. I'll activate it. Okay, so now I'm in the sourmash tutorial environment, and then I'm gonna paste the command on the tutorial. So what we are doing is that we are going to install from the channels CondaForge and BioConda, we are going to install sourmash. And so if you configure if you follow the BioConda install instructions, they will put the channel configuration, and you wouldn't even need to put both of them here. But the reason that I'm putting this is to, if I only said BioConda, it wasn't going to see the CondaForge packages. And BioConda is something that's built on top of CondaForge. Yeah. So CondaForge is a more general, they accept more software. that's not necessarily bioinformatics related. And Bioconda is the software that's only bioinformatics related. So to avoid duplicating efforts of having stuff in Bioconda and CondaForge and then getting conflicts, like they work together to say, okay, this is a like a pro package that's more general, so let's put this in CondaForge instead of keeping in Bioconda. And same thing for R, for example. There are many packages that were migrated from Bioconda to CondaForge because they are more general than just for bioinformatics.
How will you know if there's some change in the channel?
So it should be transparent. Like if something migrates from Bioconda to CondaForge, it will continue working because to use Bioconda, you should configure to have both of them. So. Okay, so now we have sourmash installed. It didn't show all the downloading packages step because I pre-downloaded the packages before, so I didn't kill the connection. But now I can say sourmash, and then I get the help message for the sourmash command. So the first thing that we are going to do is create some signatures. And a signature for sourmash is a collection of me-hash sketches. So me-hash being the original approach, a signature, it's something that you say, I have this file, I'm going to calculate me-hash sketches for different case sizes, but I'm gonna bundle it all together. So in this example, I'm going to start by downloading a genome assembly for, it's an E. coli genome. And I already have this downloaded here, but we can take a look to see that it's actually a genome and how this file looks like. Sorry, I need to go to sourmash data. Here. So this is a FASTA file that has the genomic sequences. And a FASTA file usually starts with the greater than sign here and an ID. And then after that, you have the sequences. And when you have a line break like this, it continues on the next line. So this is one long sequence. And I'm only getting the beginning of the sequence because if I do the full sequence, it's going to be kind of curious. Like, yeah, it's a lot of stuff that gets printed on the screen. So this file I had downloaded before, but if you follow the instructions here, that's what I did. So if you use curl, you get the same file. And to calculate a sourmash signature, the command is called compute. And in this case, I'm saying to compute with a case size of 21, 31, and 51, with a scale of 2000, which in the future is going to be the default, but for now we need to specify. I'll go into what scale it actually is later, but it's a variation of the basic method. This other flag, track abundance, is saying on top of hashing all the k-mers and counting if they are present or not, also count how many times they show up in the data set. So for each hash, you will also have a count saying, oh, this k-mer showed up 20 times or 1 time or 5 times. And then the show, that's going to be the output file and then the input file. So.
Why do the scale track abundance flags have 2 dashes instead of just 1 dash?
So usually when you do parameters on command line interfaces, like kind of like the defaults for Linux, is to have, if you have a short argument, it's with 1 dash. If you have a long argument, you have 2 dashes. So if you look in sourmash compute, there you can also run this as sourmash compute dash case size 21. Yeah. So 1 dash for the abbreviated one and 2 dashes for the long one. That's not completely standard. Like it's kind of a convention. It's not every single tool that follows that, but yeah, we try to follow the Linux conventions or GNU Linux. Well, anyway. let's see. So now we have this signature, and we can see the original file size for this. That's kind of annoying. I don't want to keep hearing my. Okay, so the original E. coli genome was 1.4 megabytes, which is pretty tiny actually. And our signature file is .seq. It's 129K. This one is actually compressed, so that's smaller than the uncompressed one. And the signature is not compressed here, so that it's larger. And it might not sound like that much of a savings. It's about a tenth of the original size. But with metagenomes and things that have lots of repetition, that gets a lot smaller. And so instead of having all the original data lying around when you want to do some sort of comparison, now you can keep this signature file and avoid having to recalculate a lot of stuff all the time or store all that data, which is kind of annoying. So with this signature, we might do some different things, but one comment is that if you're comparing reads against high quality sequences like an assembled genome, you might want to, well, so this part here is more about like how to deal with errors in the original sequence. So if you're comparing stuff that's high quality, like an assembled genome, you don't need to do error trimming or try to cut sequences that are not over a certain abundance. If you're comparing reads, which are things that have a lot of errors, you might need to pre-process that and trim adapters and remove errors and that sort of thing. So we will start by, again, you're going to download some raw reads. I already downloaded this file. So if we look at this one, this is something that was downloaded from the SRA. And that's why it begins with this ERR name. And this time, this is a FASTQ file. A FASTQ file is different than a FASTA file because a FASTA file only have the ID and the sequence for a genomic sequence. A FASTQ file has the ID on the first line, then the sequence, then this line here, which sometimes mirror the ID line, but it's more like a separator. And then you have the quality of each base of that sequence. So this is the sort of file that the sequencers output, and it's the things that you get out of a genome sequencing center, a genome core. And after you assemble, most assemblers only output FASTA files because it would be kind of hard to calculate what's the quality for each base on a. You can actually do that, but mostly downstream analysis are using FASTA files and they discard this quality data. In the case of sourmash, we also discard the quality data. We are worried about the sequence. So that's where you want to, you might want to do some quality trimming before, so you're giving good quality sequence to analyze for down the line. So because we are using a read here, there is another tool that the lab develops called KGmer that has a script for trimming low abundance sequences from read data sets that you get from a sequencing center. And the goal here is to get things that are that show up in reads and have very low abundance. They are very likely to be errors because if you sequence stuff, and because of how sequencing works, you usually have amplification and like you have different, you have a higher coverage than just once for each read. So what this script does is that it tries to look into each read and see what's the the coverage and what's the abundance of those bases. And then if they are too low, it's going to remove that read from the data set. So, okay, this is the output for our CageMare. So this step here, we are trying to get our reads data set closer to the quality of what would be an assembled genome without going through the genome assembly process, which is something that's sort of complicated and take a long time. So this is also something that you can do before you do genome assembly, do the stream Lowaban, because genome assemblers also suffer a lot from having too many errors, and they might start using too much memory if they have too many errors. So this is a sort of trick to also avoid having our genome assemblies taking too long to run. And yes. So depending on what you're trying to do. So for example, if you're working with metagenomes and you try to find stuff that's inside your metagenome, it might be very hard to actually assemble. And the other issue is that if you try to assemble the metagenome before doing that, you might, like the assembler might discard some information that's actually real. Because the assembly process has, it's still running, okay. The assembly process has, when you build a genome assembly graph, things that are errors, they will generate bubbles in that genome assembly graph. And so, because all the tooling for dealing with genomic data usually expects that FASTA file, that sequence of things, when you have a bubble, you have a, so you will have something like this. And this was one sequence or many sequences, and this was another one. And if the genome assembler sees this, it will try to do some sort of heuristic and decide what's the real thing. And if you say, if the genome assembler is saying the real thing is the one that has more coverage. So let's say this path here, like 10 times it goes over here, and this bottom one is only one time. The genome assembler might go and see, yeah, this one only happens one time. So that's probably an error that was introduced and created this fake path on the genome assembly graph. So I will not consider this one, and I will only take this one. And it's going to output that top as being the linear sequence, the fastest sequence that we're going to have. Turns out that for metagenomes, that's really complicated because you pretty much never sequence deep enough to have a lot of coverage around. So leaving the genome assembler to take that sort of decision might actually make you lose real biological information that you have, like real variation. And the other issue is that if you're dealing with this sort of stuff, it's rarely this size of a bubble. It's actually something that goes, it starts here, and then there's another thing here, and another here, and another there, and they go in the middle. And then the assembler is like, I don't know what to do with this, and ignores this part and continue with other places. So the benefit of not doing assembly for doing this sort of analysis and focusing on the reads is that the k-mer content of the genome assembly and the reads should be fairly similar. Like discarding errors, they should actually be the same. So if you're working with the reads, you actually have more biological information than the genome assembly that you get, or the metagenome assembly that you get after. So a lot of the things that we do in the lab, we actually try to avoid assembling and try to work with the reads. And sourmash is one step on that. Space graphics, which is another beast, which I'm not going to explain now. It's another way of trying to deal with this variation and all this complexity that happens with metagenomes, for example. Sorry, that was a long, but it did help finishing calculating the data. So now we have a trim file, and the name of the trim file is this one. This thing in front of me, it's kind of annoying. Okay. So if we look at the, let's all of them up so we can see. Okay, the original file was this one. The error trim one is a bit smaller. So the difference here is that we removed some reads that were deemed too low to be, that probably they would be errors. In this case, because this is, this data set, this is, I think this is actually reads from a single genome. It's not that big of a difference. For metagenomes, it does make a lot of difference. So let's calculate the signature for this one now. And again, same parameters as before. You're actually thinking about making these parameters the default ones, like decay and scale and track abundance, because we use this all the time and it's a lot of characters to type all the time. And while we are doing that, yeah, something that's also relevant is that this is all running on my laptop. This is not a cluster nor anything. So it's actually something that you can, it's the sort of analysis that you can do on your laptop. You don't need to submit to Farm or to something larger. Okay, so this is kind of like how we create signatures and things that we need to worry about trimming sequences and so on. Okay, this finished. Kind of curious what's the size of this one. Yeah, this one is 149K, which is way smaller than 61 megabytes. So this is more representative of the kind of how much space you save when you have when you're calculating signatures from reads. So after you create a signature, what sort of things can you do with signatures? So the first one that you can do is you can compare many signatures to each other to see like how similar they are. And in this example here, we are going to use a data set that's RNA-seq data. And this experiment, if I remember correctly, it's yeast. Is it yeast?
Yeah, Luis.
Yes.
It's yeast. And sorry, is this the Estrechedel paper? Yes. So it's yeast, and it's from an SNF2 mutant, which is a global transcriptome regulator. And so they were trying to compare sequencing, but also the biology between these mutants. So they had 647 sequencing samples that were sequenced, and so we can compare all of them really quick.
Okay, thank you. I'll say this to people because I don't think they heard. So these are 647 different samples. It's real biological samples that were prepared, and they have two different kind of samples. And Taylor just said what SNF2 was. I don't remember. It's
global transcriptome regulator.
Global transcriptome regulator. So that sounds important. So, but the thing here is that we have 647 of them. And like it's saying here, if you're going to make an MDS plot to compare how similar things are with the full data for these data sets, you need to trim the reads, download or build a reference transcriptome for these samples, then quantify the reads using a tool like Salmon, and then you're going to read the counts into R and make the MDS plot. So that's a lot of steps, and you need to have all the data available for all these things along the way. So for this example, the data is pre-calculated in this place, but it would be the same process that we use for creating the signatures using sourmash compute for each sample. Next time, not next week, in 2 weeks, we'll start talking about SnakeMake, which is a way for automating running out these commands. So you don't need to type sourmash compute for 647 of these samples. But for now, we are going to use these signatures that are already pre-calculated. And I downloaded them because it could take some time to do it. And here we have 647 files, one signature for each of the samples. So it's a bundle of me hashes. Yeah, because we have for different case, like we have one for each case size. Yeah. And you can have more stuff, like you can make a protein me hash and other things that sourmash can do, but let's not go there now because then it gets complicated quickly. But it's possible. Let's see. So the command that we are going to do to generate this plot, it's called sourmash compare. You need to specify which case size you want. So the case size here must be present in all the signatures that you pre-calculated before. But because we did 21, 31, and 51, these are all in the signatures. This is the output file that's going to be generated. And then we pass all the signatures that we want to use to calculate. So in my case, it's inside this church.sig directory. And I'm saying select every .sig file that's present in that directory. So it's going to load all the signatures. Go, laptop, go. I didn't. Let's go there. How big is that? So that's 58 megabytes of the signatures for the 647 files. So we loaded all the signatures. And so now it's going to build that a matrix that's every data set in the columns, every data set on the rows, and then for each position in the matrix is going to say how similar they are to each other. And if you're into similarity matrix, it's a symmetric matrix because the distance from one to the other is the similarity is the same. If you're using the Jacquard distance, not if you're using containment, but we'll get there.
And is that the point where you use the same one?
So the signatures are already this simple K-mer composition, like it's the small one. So the sourmash compute is doing the subsampling and saving the subsampling. So for the Jacquard similarity estimation, now we use the full subsample data set, but it's like at this point on this sourmash compare, we are not recalculating the original data to get the subsample. It's already available for us. Okay, so now it's reporting some stats saying that the minimum similarity that it found in the matrix was this one. It created a file with the labels, so the names for each data set, and then a distance matrix, which is a big NumPy array in this file. So if you if you're comfortable with NumPy, at this point you can open Python and import this NumPy matrix and plot and do all the magical things you can do. But we also have like a shortcut here, which is a plot command. And if you do sourmash plot, you pass the dash labels here means I want to put the labels in the matrix. And this is the distance matrix that was generated. So it read the matrix. It's 647 by 647. Loaded all the labels, saved an histogram of the matrix values and a dendrogram, and then the well, it's actually not the NumPy distance matrix, so we should fix this message. But now we have a PNG file, which is an image that we can actually see. Get out of the way. So this is a plot with the similarities for all these 647 samples. I'll actually go with this one because I think it's a bit bigger. So because we have that many samples, it's pretty much impossible to read what's happening here. But the sourmash plot command is more for doing this quick skimming of the data. Like if you want to do a better plot when you have that many samples, like there are better alternatives than using sourmash plot. But it's at least a start that you don't need to go write matplotlib commands to plot this data on a Jupyter notebook or something like that. And in this plot, we can see that it pretty much separates in two big chunks. There is this ocean of blue here. And the color bar here is like similarity going from 0 to 1. So this block here is pretty much very similar to each other, all these samples. And this block here is also like another cluster of similarity. And we can see with the dendrogram that's, well, there is a third one here in the bottom, but they pretty much separate and cluster together.
Can I say something about that?
Major blocks.
Well,
Yeah, there are some outliers down here. And now that you have this similarity matrix, you might choose to see like, okay, if you are working with outliers, you can extract like what are these ones and go for them. Or if you're more interested in these other big clusters, you can get only those signatures and deal with them. So a lot of what we do with data analysis in sourmash is to get this, again, name of the talk, these quick insights, where you get a kind of a summary of the big things that are happening on your data set, and then you can dig down into the specifics and then use more computationally demanding things to do your analysis. So at this point, you can get, for example, this cluster here, and because it's like, let's say it's probably around 30 data sets that are here, you can actually go and align them all against each other. And it might take some time, but it's doable versus doing 650 of them and aligning all of them. That would take a long time. I will not unmute to let Taylor talk more about that. So other use cases. This one is, we have raw RNA sequence reads, and we want to detect if there is something unexpected in our data set. So for example, let's say that you send a sample to be sequenced at the genome core, and you get your data back, and you want to do a quick check to see if everything that's there, it's what's supposed to be there, if there's no contamination with other things. So for this feature, we have pre-built, and this is something that Taylor and Tessa built. They went and downloaded all the eukaryotic RNA samples from RNA-seq and GenBank and built an index with all that data. And then they also took a data set which is marine transcriptomes and build a database for those signatures. And if I go look at that data, no. Yeah. That's not very useful. Yeah, well, these are the 3 RNA data sets. So we have this data set, which are all the eukaryote RNA transcriptomes built into GenBank and RNA-seq. There are caveats on how they build that. Like they take the genome and try to build the transcriptomes based only on the genome. They are not building a reference transcriptome out of RNA-seq reads. So it's more like a translation than a real transcriptome, but it's good enough for these purposes. The other one is the MMETSP. And finally, there is a human RNA-seq transcriptome here too that we can also use to search for contamination. So this is the 2 that I executed before. For this example, we are going to download some sequence reads from a wine fermentation sample. And this is also downloaded here. And for this example, we are going to use a command called sourmash gather. And the idea for gather is that it's going to do a sort of taxonomic breakdown of your sample. So it's going to try to find based on a database, every match that it's in that database. So if we try to execute this, I'll leave this executing while I talk. We are saying we are running sourmash gather with a K of 31 and a scale of 2000. We are saving the outputs every match that we get to this CSV file. We are passing a signature. And then you can pass multiple databases. So one benefit of this is that if you build one database for example, the samples that you have in your freezer in the lab, and you get more samples over time, you don't need to rebuild the original database. You can build another one with the new samples and then search against both of them at the same time, just by passing more and more databases here. I'll unmute here. Taylor, do you have anything else to say about this, since this was your data?
Sorry, it took me a second to. Sorry, it took me a second to unmute. Oh. Well, no. We'll see once the results start streaming across the screen. It's a wine metatranscriptome, and it should have like yeast and some other microorganisms and some grape in it.
Okay, yeah, so that's actually important. This is wine in the sense that you took everything and it's not one metatranscriptome, it's a metatranscriptome, so there are actually more stuff in there. And this part is my fault, is that it's slow. I should actually go and fix gather to be a bit faster. Sorry for that.
It'll be done very soon.
So yeah, I actually didn't introduce myself, but I mostly work in sourmash in the lab, so I do mostly of the, yes.
I have a question. To detect the contamination, so you get the standard signature from the reference?
Yeah, so the way that these databases were built, the GenBank ones, is that if you go into GenBank, they have the genome assemblies.
Yeah, I mean, if you have the genome reference, why don't you map all the grids to the genome reference and get the contamination?
Yeah, because this one is a metatranscriptome instead of a metagenome. So transcriptomes are different than the, well,
I think it's same.
you,
I
you
mean, you can get the contamination by matching the risk to the genome. If the risk can't map to the genome, it should be the contamination.
yeah, but mapping is it's more involved than doing what we are doing here. Let's say that you want to map your query, your new data set, your new sample to everything that's on GenBank. So just for building the reference database, for actually doing the mapping, that's going to be massive. Right? No. Okay, sorry, I actually lost and started printing. So these are the results that we get back from our original metatranscriptome. The first column, or the second column here, the P query, is how much of the query that we gave, the wine.sig, signature. So from that file, 79% matches this first yeast from the original data set. And then you start getting lower abundances. And this column, if you get if everything has a match, you get this back to 100%. This column, the percent of the match, is from this this match that you got from your reference database. 28% of that match is contained on your query. Yes. Yeah, because we also, the thing that we have here is based on reads. So you might not get all the matches from the original genome. And also you might have like different strains happening in this. It's very rare for you to get with your read data set exactly the original genome, because there is strains happening. And so
Louise,
yes,
this is also a transcriptome. So at this point in fermentation, only a small portion of the genome was being expressed. So 28.5% is actually pretty high.
Okay. Did everyone what Taylor said? No. Okay. So this is a transcriptome sample. So this is only showing how much was actually transcribed. So getting 28% back of the full transcriptome is actually pretty high. That's what Taylor said. So the other things that we see here, we see wine grape, which is good because there was grape inside the wine fermentation. More yeast. I don't know what's this one, but there is this one in the middle, which is house mouse in that sample. And that's not the sort of thing that you want to see on a fermentation. And this actually is not something that there wasn't a mouse that actually fell into the fermentation. What happened here is that this was contamination on the sequencing center. That when you put the samples to sequence, you might mix a bit of the samples in the lane in the flow cells, in the lanes of the flow cell. So we might end up getting some contamination over that. And then at this point, you can decide like, okay, I see that I have mouse here. Do I want to process this data and remove things that match to mouse and work with the rest of the data? Or do I want to go back to the sequencing core and ask them to resequence this because there's actually contamination happening and there shouldn't be? So yeah, no human in this one. And yes.
I don't know if this is like a silly question, but how do you know that it's
Because this was this sample is something that Taylor worked on, and then she asked the sequencing center, and they actually sequenced mouse together with her sample. So yeah, I'm not saying like this is, again, sourmash is very good for giving you insights or generating hypotheses, but you do need to check them. Like you can see something like this and say, yeah, there was contamination. Like there's something fishy going on here, but you need to go and investigate what's actually happening. But the alternative would be like you never find this, and then you start calling mice genes out of your analysis further down the line. So it's better to have this now instead of in the end of your pipeline. And I think we're gonna cut the presentation here because it's getting quite long, and we actually want to have the questions later for the med session. But all the other examples here you can download and run the commands. Yeah, this is the mouse example. And we have some other examples of comparing reads to assemblies. Building a database for, in this case, it's using E. coli genomes and searching against specific set of signatures. And in sourmash, if you go to the download page, like we have some prepared databases for RNA-seq and GenBank, which are somewhat old now. We are in the process of updating this, but we already downloaded like the full GenBank and calculated signatures and put it in a format that's fast to search against. That's about 100,000 genomes that we have for archaea, bacteria, and fungi. Yeah, and then the other example is to do taxonomic classification on metagenomes, which is also using Gather, but you can also use to search what are all the things that are present on your metagenome. So yeah, sourmash. Mm-hmm.
Do you take care to balance your database? Like, so for example, I have this giant culture,
Mm-hmm.
but the thing is, culture
Mm-hmm.
in order to use sourmash, should I strip those down so I have more of an even representation genus, or is it fine?
So the gather command was actually created for dealing with that problem. So the original command for searching data was sourmash search. And the issue that we were having when we had a metagenome and we wanted to search what was inside that metagenome is that if you had one E. coli match, you had actually 300 E. coli matches. And then it was very hard to find out the different things because there were so many E. coli that were appearing in your results. So what sourmash search does is that you're trying to answer the question of like giving a database how many similar things, like over 90% similar things I have in the database compared to my original sample. So this is good for things where you have a single genome or something that's one thing, not a metagenome. The process for Gather is that instead of searching for everything that's over a certain similarity threshold, it's going to search and find the best match for your metagenome. And that might be one E. coli. And then from your original signature, it's going to remove that E. coli sample and make a smaller signature, and then search against for the next best match and keep doing that. So you might, if you have very specific E. coli strains, for example, you might get more than one E. coli match. But the benefit is that you get a result that looks more like this, saying, okay, you have this species and 4 other equal matches for this species, instead of having all of them being printed here. And so it's better to do the breakdown on what's on your metagenome than search. So depend on your application. Like if you have a metagenome or a metatranscriptome, usually Gather is the best approach, like what you would want to use. If you have a single genome, then search probably is more useful for you. because you can find all the other similar things that are available in public databases or your sample collection.